Oscar, il y a 238 slides. On n'est pas couché ce soir, je pense. Hein.
C'est pour ça que je t'ai dit de ne pas les regarder avant, parce que c'est des animations.
Ça fait peur, ouais.
Non, mais ça, c'est juste la première partie.
Ah oui, oui, d'accord.
Après.
Je me disais.
Bonjour Oscar.
Bonjour, Caroline, bonjour.
Bonjour Caroline.
Et bonjour à tous, oui, bonjour aux personnes qui sont connectées.
Bonjour.
Bonjour.
Bonjour Jocelyne.
Bonjour Céline.
Bonjour à tous et à toutes.
Bonjour à tous. Je vois que c'est
Donc, on est une fois de comment les collègues nous voient des profils. Sauf que c'est un enfant. Non, parce que j'ai vu, il y a forcément pour ça, il n'y a pas la caméra. Ah oui, d'accord.
Oui, te vois. Te vois là-bas.
Voilà. Donc, bonjour à tous, bienvenue. Hello everyone. So we'll begin in a few minutes. We are waiting for Renaud to come over here, and we can begin then, I think.
Ah, c'est Renaud qui manque. Oui. Peut-être, oui. Je vais appeler Renaud parce que sinon.
Pronto.
Pronto? No, tu nel momento in cui arrivavi mi dovevi chiamare. Va bene. Va bene. Va bene. Ok. Ok, adesso vabbè, io adesso devo attaccare che sono in una commissione. Ok? Ciao 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 ciao. Ciao.
But I don't know whether you can use. I'm checking my sound, my camera, my micro. It's okay for everyone. So okay, perfect. We are. Reno is here, so it's okay. Mireille. Mireille, si tu veux venir lire ici, c'est bon? Ouais. Salutez-le, tu les as pas ici.
So welcome to all, and thank you to the members of the jury to be connected at time. And thank you to Renaud. So we are happy to be here today for the presentation of the defense for habilitation to direct research of Mr. Oscar Acosta. I would like to thank also the members of the jury to be in confidence with me to represent the president today of the jury. This jury is multidisciplinary, international, and as you can see, with a very good gender balance. So I will introduce the members of the jury. Jocelyne Trocaz, research director, CNRS at laboratory TILSE IMAG from the University Grenoble-Alpes.
Hello, everyone.
So Jocelyne works in the field of image processing and medical robotics for computer-assisted surgery, and she's reviewer of this work. Professor Konstantinos Marias, so associate professor at the Department of Electrical and Computer Engineering at the Hellenic Mediterranean University in Greece. He's director of the Institute of Computer Science of the Foundation for Research and Technology, Hellas, in Greece. He works in medical imaging processing and is a reviewer of this work. David Samu, research director, CNRS, at laboratory Creatis Sémignon from the Cancer Center Léon Bernard. And so David Samu works on nuclear imaging and image-guided radiation therapy, and is a reviewer of this work.
Hello.
Laura Cella, research director at the Institute for Biostructure and Bioimages from the National Council of Research in Naples, Italy. She's also consultant medical physicist at the University Hospital of Naples. And her research area concerns the physics of radiation oncology. Caroline Petitjean, she's an assistant professor rehabilitation to direct research from the laboratory LITIS, University of Rouen, and she works on image processing and machine learning classification with application in medical imaging. So Renaud de Courvoisier, here present, professor and hospital practitioner at the University of Rennes, Rouen, director of the Cancer Center at Jean-Marc in Rennes, where he's developing expertise in prostate cancer and high precision irradiation techniques. He is member of the LTSI and member of the faculty team in the lab. And for me, I'm Michel Garot, so professor from the University of Rennes, member of the LTSI and member of the faculty team. And I work on medical image analysis side for the planning and assistance of cardiac interventional procedures. So now we will begin the presentation. So Oscar will present his work with about 40 minutes.
Thank you, Mireille. Thank you for this nice introduction, this fine introduction. Thank you for the members of the jury to be here. Thank you to the reviewers for time spent reviewing my manuscript. And thank you, Renaud, to be here. Thank you to the audience. And I know that there are some people as well in YouTube following in Colombia as well. So hi everyone, my family and my friends all around the world. And well, thank you for this kind of opportunity to present this work, which is entitled Towards Personalized Planning Prostate Cancer Radiotherapy, which is the work I've been developing in the last few years in the lab. So first of all, this presentation will follow this structure. I start with my CV, some clinical context of my research activities, the research project, and finally some conclusions. So first of all, I am an electrical engineer from the University of Los Andes in Bogota, Colombia, where I also did a master's in biomedical engineering in 1998. I first arrived to France in 1998, as well as an internship to the Telecom Bretagne in Brest. Then, after some back and forth in between Colombia, where I worked as a commercial engineer and electrical engineer in Colombia, I finished my PhD in France in the University of Rennes in the LTSI in 2004. Afterwards, I went to Australia, when I worked in 2005 as a postdoctoral fellow at Westmead Hospital in Sydney. And then between 2005 and 2009, I worked as a research scientist developing biomarkers from MRI to diagnose Alzheimer's disease. This work was a research scientist in the CSIRO Health Research Center. Then I took a position as associate professor, maître de conférence, here in Rennes, the University of Rennes, where I devote my teaching activities to signal and image processing, computer vision, computer science, mathematical tools for engineers, etc. And since 2017 until this year, I was the coordinator of the Masters in Electronics, Electrical Engineering and Control Systems in the ESTIC department. Concerning the research activities that are devoted to the exploration of multimodal data for prediction of response to radiation therapy, namely toxicity and recurrence. And this was work that's been developed in the IMPACT team. And now I am affiliated also to the LTSI, but also to the Centre Jean-Marc here in Rennes. And the main clinical target of this works has been around prostate cancer, and is the following things that I'm trying to show you today, which constitutes the works in these last years. So first, the clinical context. The prostate cancer is among the most common type of male cancer in developed countries, with almost 10,000 deaths in France. But it's roughly out of 6 men will be diagnosed with prostate cancer. Once the cancer is diagnosed, either by digital rectal examination or blood test or even histology, there are different treatments that can be proposed to the patients. This treatment can be either systemic, like chemotherapy or chemotherapy or immunotherapy, but also it can be local, like a prostatectomy, surgery, radiotherapy, or HIFU. Two kinds of, well, the interest of the radiotherapy is that it can be applied to localized tumors, and the interest is that it preserves the organ. It can be used also as a secondary treatment. So two kinds of radiotherapy can be distinguished. The first one can be brachytherapy, where there are some seeds implanted within the prostate, and external beam radiotherapy. My works have been all around the external beam radiotherapy. Well, the principle of radiotherapy is to use radiation in order to destroy the malignant cells. So the beams that are delivered by an accelerator from the external body are maximized. The idea is to maximize the dose of the energy delivered to the target, which arises when these beams intersect. Well, I try to explain the workflow, typical workflow used nowadays in radiotherapy. There are two big steps. The first step is the planning, and the second step is the irradiation. Well, what happened in the first step? We have a CT scan, which is acquired in order to plan the treatment. On this CT scan, there is a segmentation, usually manual, of the prostate and organs at risk, like the rectum, the bladder, etc. There is the PTV, which is defined, is the prostate target volume, which is the prostate plus some margins in order to have problems of uncertainties of displacement or deformation of the patient. And in a third step, there is a plan, which is plan. What is the idea of this plan? Is to simulate and propose some beam configuration in order to ensure that the delivered dose is the same as the planned dose. This in this step, dose distribution is computed. This dose distribution is being validated by radiation oncologists based on some constraints. Usually, there are constraints based on the DVH. What is a DVH? It's a dose volume histogram. This concept is very important here to be taken into account, because from this concept, we work in order to propose new methods for prediction of toxicity. So what is the idea? The idea is you have a volume, and this volume, histogram represents the certain amount of dose that is delivered or this on a certain amount of volume. The contrasts are based on this DVH. For example, we may say, following some recommendation, that the 95% of the PTV should be irradiated with 95% of the prescribed dose. In the organs at risk, such as the bladder and the rectum, the idea is the opposite. The idea is to decrease the dose that is going to be delivered to the organs. So the recommendations say, for example, that the maximal dose, which you have it here in the x-axis, should be less than 76 gray, for example, and that the 20% of the volume here should be less than 72 grays. So based on these constraints, on these dose volume histograms, the plan is validated, and then we proceed to the second step. The second step is the irradiation. What happened? The patient is located in the linear accelerator. So it's a localization either based on tattoos or CBCT or image-guided radiotherapy. And then the dose is delivered using accelerators like with technology like MRT, or more performing like CyberKnife, or today the MRI linear. This treatment is not delivered only in one treatment, in one single fraction. It's delivered in several fractions. So it's a fractionated treatment. Usually, in the prostate cancer, we have 2 grays per fraction, from 1 to 40 days. So accumulating 80 grays at the end of the treatment. But the trend today is go to well, there's some evidence showing that when we go towards hyperfractionated treatments, the local control is improved. So in conclusion of this first part, we might say that the dose should be maximized when these beams intersect in the target, but at the same time to decrease the dose to the surrounding organ at risk in order to, so it's a compromise between the decrease of secondary effects, decrease of dose to the organs at risk, but also to increase the dose to the target in order to achieve local control. The toxicity is one of the secondary effects, and the recurrence is the loss of local control. What is toxicity? Toxicity might affect the sexual function, the urinary function, or the rectal function, and the recurrence might arise in a non-neglectable number of cases. It can be up to 40% in the population of high risk. And the toxicity is very important. So one of the challenges in radiotherapy is how to predict the toxicity, how to predict recurrence in order to improve the treatment at the planning step. So one of the issues, and is the center of my works in this last year, is how to propose methods to personalize the planning, to personalize the treatment by answering to 2 key questions. The first one is the prediction, how to predict the toxicity, how to predict recurrence, and how to propose methods in order to improve the description of the target and organ at risk. All these questions in a context of technological developments, huge technological developments in terms of accelerators, but also imaging devices, in terms of methods, big data methods, image processing, computational methods, etc. So I try to explain or to divide these works in 3 different axes. The first one is the axis of, well, with the goal of prediction of personalized planning. The first axis is the prediction of toxicity, prediction of recurrence. And the second axis is MRI planning in order to improve the target. All of this, with taking into account, we have a huge amount of data that have been gathered or have been collected during the whole radiotherapy workflow. This data can be clinical, biological tests, the planning 3D dot distributions, the imaging, or even the histopathology. So the idea is, in a context of big data, trying to use the data in order to answer to this question. That's the first thing. But the second thing is, in the data are not enough. So how to produce more knowledge from radiobiological considerations in order to answer to this question. And around this question is the methodological developments I'm trying to explain in the next few slides. So answering to 2 questions. The first one is how to display the data and how to introduce knowledge in order to answer to this question. So the methodological contributions can be split in different boxes that you can see here. And well, I'm trying to develop each one of them in the next slides. So the first one is to the question of toxicity. The question of toxicity has been tackled in several ways. The first thing, we have to understand what models have been proposed today. So what is the idea? The idea is to go to the full exploitation of 3D dose distributions in order to answer, to see whether we can predict the toxicity events. Well, traditionally, what has been used today is that the whole data are not fully exploited. What is only exploited today is only the 3D dose distribution, as you can see here, via the dose volume histograms. So most of the approaches that are very well known in the literature are based on the distribution that is considered the whole organ. This is the most popular model, is the NTCP, or normal tissue complication probability model, which establishes that, or which proposes the hypothesis that the more the organ is irradiated, the higher is the probability of being of suffering from toxicity. So this is a sigmoid that is fitted from a population, and we determine some parameters to a single population. Well, the literature has been populated with different methods, and when we started doing these models, we proposed it in the first PhD student that I co-supervised, it was Juan David Ospina. We proposed machine learning methods, random forest particularly, in order to answer to the question, to see, we investigated the question whether we will be able to improve the prediction compared to the traditional NTCP models. And also in inter-team work in the lab with using the skills developed in the lab in ICA, Independent Component Analysis. Aurélien Fargias has proposed also methods in order to explore the data to improve the prediction. The problem of these methods is that they only use the DVH and doesn't take into account the potential spatial relationship that might exist between the dose and the toxicity effects. This, to a larger extent, this represents a very big problem because the DVH has several drawbacks. The first one is that, of course, the DVH is a very reduced representation of the 3D plant dose distribution, and the toxicity might involve the several structures at different spatial scales that are not taken into account in DVH models. Also, the different 3D dose distribution may conduct to or may lead to the same DVH, and mainly the information, this spatial distribution is completely lost. So any local correlation can be established between the organ that can be affected by the dose and the secondary effect. So in order to answer to this question or get around this question, several approaches have been found in the literature. Most of them at the beginning of the 2004, 2005, trying to propose methods dividing the organs in different sub-organs and to use also the DVH in order to see whether by a small portion of the organs we can be more predictive. This was, it's not enough. The dose surface maps was also proposed in order to try to explain in a single plane the dose distributions. leading to this kind of maps relating the difference between a population with toxicity and without toxicity using this dose map. The problem with these approaches are they are limited to organ surfaces, and of course they don't express they doesn't take into account the involvement of multiple structures in toxicity. So that is the core of our contribution, is to propose methods in order to explore the 3D dose distribution in full. So the idea was to propose Voxel-wise methods, and this was first explored in Gael Drean's PhD. The idea was the following: we have a population of patients with toxicity and without toxicity. We select one of the templates, one anatomical template, as a single coordinate system, and then by doing registration, non-rigid registration, the idea is to bring the population to the same coordinate system, to apply this deformation to the dose distribution as well. So in the same coordinate system, we can refer to the same anatomical points when we are referring to when we study the anatomical template. So the dose can be comparable in the same anatomical space. So this led us to propose this workflow that you can see here, where you have the population, you have a template, you select the template, you do a spatial normalization via non-rigid registration. I can show you an example here. So you have the patient, you have the template. By doing non-rigid registration, you deform the patient, then you extract the deformation field. This deformation field is applied to the dose distribution, and then the dose distribution is mapped into the single template. This is the whole workflow of that. You have dose mapping, and at the end, you can do whatever you want with the voxels in the same space. So this is the first seminal paper in 2013 that's been highly cited, which proposed this method using non-rigid registration. And the idea is that you can produce a cartography in 3D. And in this 3D, you can do, for example, statistical tests to see which voxels are statistically different, statistically significant, or where this difference is statistically significant. And you end up having some regions, subregions, that may represent a high correlated event between the dose distribution of the irradiated dose and the toxicity events. Well, this paper was very nice, but the problem is that it uses a reduced number of patients. The registration method was not optimized, and the improvement in prediction was not demonstrated. So, well, we played around also with Voxel-wise method. We proposed different methods to exploit the data distribution in the same space using different strategies. But Gael Drean tackled the problem of interindividual variability, because this is a problem where you have a high interindividual variability and you can rely on a single coordinate system. So what he did is a nice study when he selected different geometries as an exemplar in different clusters, and he reproduced this Voxel-wise analysis in these different clusters to show that indeed there was a huge variability in the subregions that show up when you are reproducing this Voxel-wise analysis. So in order to cope to get around this problem, he worked in proposing a customized non-registration method based on the Laplacian distance map, which is a normalized description of the structure, and in order to have a reliable registration method that allows you to map in a reliable way the dose distribution. So using this method, he repeated the process of a Voxel-wise analysis on 118 patients to end up having this region. And this region is particularly, he showed that it's particularly easy to find in any patient. And this region is more predictive than any other split of the rectum using either the whole volume, the anterior part. And it's particularly interesting because it takes the lower part, the lower third of the rectum is rather anterior as well. So it's related with some physiopathological events that can be explainable as well. So concerning this is the rectal toxicity. Concerning the urinary toxicity as well, well, it's a more complicated, far more complicated problem because the large interfraction deformation, because the different symptoms and grades, and because the involvement of different structures. So to tackle the first question about the deformation, well, Richard Rios came to propose a machine learning method in order to learn bladder deformation from CBCT data. And actually, basically what he showed that is, although there is a huge deformation of the bladder in the posterior part, the base of the bladder is rather fixed. So his method didn't improve the prediction of toxicity. So Evgenia Milona came after, and what she nicely performed was to propose a comprehensive Voxel-wise analysis, which included a lot of different things. This approach, you see the core of the Voxel-wise analysis was also the registration part, by performing a customized registration part, but also she proposed a method for the toxicity prediction to show whether the regions are more predictive or not, and also a very nice atlas-based method for segmenting the intraprostatic urethra in order to be included within the whole analysis. What she found afterwards was that 5 regions were found as predictive, more predictive than the whole bladder for 5 symptoms: incontinence, retention, dysuria, and hematuria, retention late and acute. And the important involvement in the of the urethra in the incontinence. This work then completed the whole picture of the whole reference of the whole bibliography in terms of predictive models using those. And we populated this with all this part of based on non-rigid registration. We worked beyond the DVH. We show how to exploit the data in order to find subregions that can be used in the planning. Now the question to answer is whether how to use this subregions concept in order to go to the clinics, how to bring these subregions to the bed. Well, this has to go through 2 different questions. The first question is whether these subregions are valid, externally validated in other populations, in different cohorts. And the second thing is whether we are technically able to do it, where we are technically able to transfer these subregions to the specific patient. In order to tackle the first question, well, Evgenia Milona went to Italy for a couple of weeks, and she worked with them in order to show whether there is correspondences between the dose surface maps and the dose value maps with the same population to see whether the regions coincide between the 2 methods. And she showed that indeed there are some regions, three regions were found as correspondence, giving a clear advantage to the Voxel-wise analysis, because the Voxel-wise analysis explored the 3D dose distribution in a whole. And the second work that she went to Australia in a collaboration we established with University of Western Australia, and she performed an analysis on 476 patients from the Radiotherapy trial. These patients were treated with confirmed radiotherapy. And actually what she showed is that 3 out of the 5 regions were more predictive than the whole bladder, which validated 3 of the regions that we found before using IMRT. And well, unfortunately, the rectal subregion was not validated, but she indeed published a very nice paper in the Red Journal. And the second question about how to bring these subregions to the planning. In order to answer to this question, well, thanks to a project, Interlabics project, Comin Labs and CAMI, we developed a tool whose goal is to import these subregions into the TPS. So, for example, here you have once you have the delineated organs, you can import this rectal subregion into a specific planning, a specific patient, and then you can either plan with the subregions or without the subregion. And well, this is a very nice tool because it shows that you can push forward the dose distribution, but keeping the constraints on the PTV. And pushing forward or using this method in a nice study performed by Caroline Lafont and Anaïs Barato, they developed a pilot study on 60 patients where different planning dose distribution were performed, and what they show is that by using this subregion while keeping the constraints to the PTV, we can achieve a reduction of rectal toxicity that is around 20%. Very nice result, and this is one of the bases for the project that we are proposing afterwards, an European project that we have several partners in Europe. So as a conclusion of this part, we can say that we have proposed a framework for study the 3D toxicity includes non-read registration. It lists or yields some subregions of explored subregions. These subregions improves the prediction, and these subregions can also be used during the planning step. Well, there are some questions still to be answered, but we are working on it. The main question is the reproducibility, and the dose is not the only responsible of toxicity. There is also the radiobiology. So we are exploring this question as well. So as a conclusion, we have a scope supervising 5 PhD students, more than 10 masters, 4 postdocs, and engineer. These works have been funded by several grants, Comin Labs, CAMI, IRESP. We have a maturation project in now running. And more importantly, well, this year we had a grant, European grant, via ERA Permet, and we are in order to answer to these questions. In terms of scientific production, we have produced more than 10 journal papers in this work, more 20 conference papers, software tools, a maturation project. We have been invited to talk in ISTROS, SFRO, SPM, and we have built an international networking with Australia, Italy, and Spain. Concerning the second question about the recurrence, well, we have tackled this problem using 2 approaches. The first one is based on data. We have found predictors from quantitative analysis of medical images. This is the idea of radiomics. So what I'm going to show, although we have developed methods in order to explore the MR data and the PET image data, thanks to the works of Pierre Fontaine in the last PhD, who is finishing in October. But Federico Amador and Ricardo Gutierrez have developed methods before in order to explore data, explore MRI data, and to build predictors. So this study was performed by CameraNet. The idea was to use multiparametric MRI, this multiparametric MRI, to answer to the question of recurrence, biochemical recurrence at 5 years. So the idea was to segment then the MRI. Once the MRI are segmented, we extract features. These features, well, are radiomic features. They are standardized as well. First order, second order, texture features, geometric features, radiant, frequency, Gabor filters, etc., ending up having hundreds of features, and then to use these features as a predictor. So we build models using SVM or random forest or other machine learning techniques. And we published a paper, a very highly cited paper now, which showed that 5 features are predictors for recurrence. And the problem with approaches, which are only based on data, is that they are not interpretable. What does it mean? That the Haralick texture at 35 degrees of the entropy is related with recurrence. So it's very hard to explain how these features are related with the physiology of the tumors. And the problem is also that there's a large amount of population data required in order to answer to this question. We are facing also unbalanced data. And the problem of very often these models are population dependent. They cannot be generalizable to other populations. So in order to cope with this problem, we are trying to answer to this question by developing models based on radiobiological consideration. So this is a nice work that's been developed in collaboration, strong collaboration with the Sepia team in the lab. That was started by Viviane Aubert as a postdoctoral fellow in a local Canceropol Grant West grant. And it's been nicely exploited and moving very forward by Carlos Sosa Marrero, who defended his PhD 2 days ago. And the idea was to try to give an explanation to the recurrence and trying to explore and simulate different tumor configurations and to try to improve the personalized planning by using this simulation. So what is the idea? The idea is to build a virtual tissue containing 5 types of cells. These cells can be healthy cells, tumor endothelial cells, or dead cells. and to simulate the radiobiology involved in the mechanism of interaction between the cells. So we have oxygenation, where endothelial cells are giving the oxygen to the whole tissue. We have simulated the division of tumor cells that makes that the tumor is growing. We have simulated the resorption of the tumor by healthy cells, and the VEGF, which is a protein favoring the angiogenesis. So the idea here is, the interesting part here is that these events or these phenomena are in different time scales. We are thinking on oxygenation, milliseconds, and also days or hours in terms of cell division. And we used a library that has been developed in the lab, which is called M2SL, which allows to integrate in a single framework all these different formalisms at different time scales and spatial scales. And this is how the model looked like. Well, it's a bit tricky, a bit complicated. I can come back to if you want, but we simulated different cell cycles. And the problem with this, we simulated how the tumor responds to the radiotherapy as well. And the tricky thing with that is that it's a huge amount of parameters that are involved in this simulation. And there are problems of observability, of identification as well. So the idea is then how to reduce the parameters, a typical problem in modeling. So thanks to methodology developed in the lab as well, using screening methods. While Carlos ran more than 70,000 simulations in order to come up with a reduced model, which is supposed to be equivalent to the first one, which has only 18 parameters and involves only 2 out of 4 different mechanisms previously described. So the idea was to have here the full simulation. You have the irradiation. In the blue curve, you see the number of cells as the radiation goes. After the radiation stops, then the tumor starts growing again. And you can see here in the in silico model to the left how the tumor is growing up and the place is taken by the dead cells. So different nice thing is that different fractionation can be simulated as well. And the interesting study that was performed between Carlos and Pierre Fontaine was that we compare the radiomics, the prediction given by radiomics, namely by images, to the prediction given by the tumor, by the digital tumor. So the idea was from each individual, we built its avatar, we built a digital patient, and this digital patient was used to simulate a typical radiotherapy protocol. So we look at after a while the response of the tumor, and we compare the predictors obtained from the image with the predictors obtained by the prediction of the model. And we showed that although the number of data is very low, we obtained a very nice prediction. So the prediction obtained with the model outperformed the prediction obtained with the image. Well, this is to say that M2SL allows for this integration of multiple formalisms and in multiple scales. The stride sensitivity analysis allows to reduce the number of parameters and obtain a simplified version, but it's still some problems with observability. And in conclusion, although there is a good prediction with radiomics, the model-based outperforms the prediction and it gives explainability to what is going on with the model. So it opens the road for very, very nice developments in terms of planning, of personalized planning. The problem is still of identification of parameters and observability. And well, the response of the tumor is rather complex. So we haven't taken into account also other considerations. We have published quite a number of papers as well. We have international collaboration mainly with Chile. While Carlos was supposed to go to Chile last year, unfortunately, because of the situation, he didn't go. But still, the collaboration is still there is huge opportunities to continue developing here. And going to the last part, to the last but not the least, is we are trying to answer to the question how to introduce the MR or in order to better achieve a better targeting or to achieve a better to have a better dose planning distribution. This can be done by 2 different methodologies. The first one is the fusion of images, multimodal fusion, and the second one is the generation of synthetic CT. So what is the issues with the CT? As you can see here, well, there is a poor soft tissue contrast with the CT that is used in the workflow. There is a high inter-observer delineation variability. In the CT, there is a prostate volume overestimation. The tumor is not visible, and the tumor aggressivity cannot be assessed. So in contrast, the MRI is very rich in information. We have nowadays access to the multiparametric MRI, which gives the possibility to have access to a lot of information. And this, well, by exploring this, combining these modalities, anatomic, metabolic, and functional information can be obtained via exploitation of the multiparametric MRI. We explored that during the PhD of Frédéric Commander in a co-directed work with Antoine Simon in the lab, in trying to answer to the question on how to introduce the MRI within the planning. So this can be achieved, or we achieved that following two strategies. The first one is via registration. So we first characterize the MRI, we extract the usual information, and we transfer that to the CT via registration. And it's what we did. So we worked on methods for tumor detection, automatic tumor detection, aggressivity prediction. We were able to predict lesion score, and also in trying to target, to better delineate tumors and to better delineate prostate by using the same workflow as radiomics, but not for studying a population, but for studying a single patient. So for a single patient, we were able to build probability maps. In this probability maps of tumor aggressivity, and this tumor aggressivity was these tumor maps or probability maps of presence of tumors were obtained. This data was, or these methods were validated with histology analysis on a reduced number of patients, whereas some patients treated by prostatectomy or with prostatectomy were studied. We registered the histology to the MRI in order to see whether the markers or the features from the MRI coincide with the markers or the stains in given by the histopathology, in order to build a model that learns from histology as well to produce the Gleason score. Well, this is a preliminary study, hasn't been published because, well, we published the registration part, this because of the reduced number of patients, and so it's a in progress work. And the second step is the registration. So for performing registration between the MR and the CT, we propose an original method that was also published based on the registration of the contour registration. So we build probability maps, and we try to build a new cost function based on collinearity of the contours, and this is well, a work that was published a few years ago in 2017 in the International Journal of Biomedical and Health Informatics. So the problem of this workflow is that the difference between the CT and the MR prostate size, the computational time. And one of the big issues is that the dose distribution is still computed on the CT scans. So in order to get around to this problem, or to propose different methods in order to compute the dose plan directly from the CT, well, we worked with Axel Largent in a co-directed work with Jean-Claude Nunes in the idea of from the MR directly trying to produce a dose plan distribution going through the idea of generating a pseudo CT. So what is a pseudo CT? A pseudo CT is a CT that is generated as a synthetic CT from the MRI. So this is a co-directed work with the participation of course of Anaïs Barato, medical physicist who was doing a PhD at the same time as Axel. And in a strong collaboration with well, my former lab in Australia with Jason Dolin, who were over there, they proposed atlas-based methods to answer to the same question. So they, from an MR, they produced a CT. So Axel was developing different methods, patch-based methods with machine learning, and he explored deep learning using mainly GAN, so generative adversarial networks. to produce pseudo CTs from MR images. And they also validated the different dose distributions and came up with two nice papers showing the advantages of using GAN networks in order to generate CT, which allows also as well to produce dose plan distributions into the planning. The problem with that is we need methods for the quality control of the CT to see whether the CT is able to be used in the planning. So this is being tackled in a PhD currently going on, who is Hilda Churak, who is in collaboration with Australia, whose problem is to try to figure out or to work out the methods in order to show whether the pseudo CT methods are performant or not, and to see the heterogeneity in terms of errors through the 3D volumes. So this work is going to be, well, Hilda is going to try to Australia next September in order to continue to this work, and she's opening the roads to quality control methods as well. She's using a Voxel-wise analysis as well. So the idea is from a population trying to register them together and to analyze in a single time template the whole errors. So as a conclusion of this last part of the work, we have proposed methods for integrating the MRI within the planning workflow. The roads are open for integrating the MRI already. We are in a context where here in REN, we are going to have the MRI LINAC. So we are getting ready to that. They are based upon machine learning methodologies, and well, these methodologies are part of the skills in the lab. And now, well, there are still a lot of issues or works in terms of quality control and multiscenario studies in order to transfer that to the clinics. Well, several PhD students, postdoc, it's also teamwork, and Antoine Simon and Jean-Claude Nunes have been collaborating or working. We are working together in these problems, and we have produced a couple of journal papers, conference papers, international, and working important with Australia. And well, that covers roughly the first part, which is the previous works. Now with the project, the second part of the project. So we have built evidence of toxicity methods or toxicity and recurrence. We have shown ways of how to build accurate toxicity models and recurrence models, and we have shown methods in order to define subregions within the targets and within the organs at risk. How to use that in the project. So the idea is we have the MRI, which is bringing a lot of information. So we are trying to answer to two different questions. The first question is in scope of personalized treatment planning. The first thing is how to stratify the patient, how to determine whether a patient belongs to a group, to a determined group or not, and to see where we can classify him to belong to a particular, or to give him a particular treatment. And that's the first question. And the second question is, once he's stratified, how to target, how to tailor for him the best treatment in terms of definition of or delineation of tumor and definition of subregions of organs at risk. So in order to answer to the first question of stratification, we are going to work on the improved prediction using not only dosimetry data, but also multimodal data coming from radiobiology. We are working in terms of a recurrence model, not only in improving radiomics workflow, but also in the in silico simulations, as I showed before. And in order to answer to the second question, we are exploiting the subregion definition that we have proposed before. We are using the recurrence model as well. We are using customized target using the MRI. So the MRI becomes very important in the definition of the target and the definition of these subregions. I wanted to show you just a single example of this part, which is the toxicity model, how we are going to use the toxicity model for that. For the moment, so far, we have defined, we have proposed methods that were able to extract subregions rectal and urinary subregions that are more related with toxicity. But this was done only within a particular context. This context was IMRT, 2-grade perfraction, and only involving prostate as seminal vesicles. But we have to go further. So to go further, well, we have proposed methods in order to explore to a larger extent these models, try to explore to another different configuration, and trying to bring this subregions concept to the clinics, to implement tools in order to bring it to the clinics. So we have this year, well, last year, funds from the ERA PERMED project, with €1 million with 6 partners. It's a project that I am coordinating. And the idea is to do that, to bring these models from the model development to the clinics. So with the Spanish partner, using deep learning model methods in order to segment the subregions. And with the Italian partners, who are the leaders in toxicity, well, in the world, the idea is to bring that to a particular patient. So this is the whole work of the project. Let me skim through very quickly. So the idea is to have the subregions. We need to externally validate the spatial models. We need to automatically segment these subregions and the organ surface as well. But also we have to develop new voxel-based model. That's our part with the Spanish. Then in Italy, they're going to see which constraints have to be applied to these subregions in order to decrease toxicities. And then at the end, propose all these tools to plan for a particular patient, a particular subregion division, which is supposed to decrease toxicity. Just to show you a picture of the data, that's our works. But now we have to explore more fractionation schemes. We have to explore 2 grades, 3 grades per fraction, and going up to in SBRT and up to 6-grade perfraction. No, any model exists here. So the idea is to see whether these models are still valid in other configurations. We explore only prostate and seminal vesicles, but we have to explore different anatomies. We have to explore in case of prostatectomies, for example. So for that, we are in the European project. We are gathering data and gathering with the Italians and the local data. We are thinking and having more than probably 1,000 patients in order to answer to this question. So we are at the beginning of the project, and we are still well gathering the data. And the second part that I wanted to really reinforce is to adapt the therapy by using this concept of digital patient. The idea is to have, for an individual patient, has his avatar, and on this avatar we can perform every kind of simulations, and we can bring this patient from the area of risk to the area of no risk, or the safe area, for this patient, tailor his treatment in order to decrease the risk of prediction. That is the second aspect I wanted to develop here. Well, as a conclusion, final conclusion of all this work, we developed methodologies in image processing, population analysis, voxel-based, machine learning, modeling, in silico simulation, in order to answer to the question of personalized radiotherapy in terms of prediction of toxicity, prediction of recurrence, to improve the target and organ delineations, and to perform those planning from the MRI. To do that, well, it's not my work, it's the work of a whole team, of a bunch of nice people I've been working with, 13 PhD students, engineers, postdocs. It is a fruitful scientific production with 2 book chapters, 52 peer-reviewed international journal papers, where we have published more than 40 peer-reviewed conference proceedings in both in methodological aspects, but also clinics. MICAI, ISB, MBS, BI, SPI. We have been organizing invited sessions in the conference like IEEE and NBC. We have involved in transfer activities. We have a patent on the part of the toxicity part, which is part of the European project. We have developed tools, maturation projects, large international collaboration with Australia, with Italy, Spain, and Colombia as well. And well, we have, thanks to these grants, national grants, and now the European grants that allowed us to work, to hire people, to get people working together. And well, these things are, I think, after these 10 years of work, I think things are just starting. Just to finish, I've been animating a lot of different activities other than scientific here. This is a picture of the summer school I am organizing with other nice people. Jean-Louis Dilenseyer, who is here in the room, is part of this organization of this international summer school, which happens every 2 years in Saint-Jacques-de-la-Mer here in France. And we are very excited because we have the bid to organize IEEE ISB 2023 in Cartagena de Indias in Colombia. So I'm co-chairing. I have the opportunity to co-share this conference with Natasha Lepoe and well, my friends in Australia, Olivier Salvado and Enrico Grisan. And there are other person in the lab who is John Baxter, who will be part of the organization of this nice conference in Colombia. So with that, I wanted to thank all the people who, well, it's not mentioned here, but I've been working with in the last few years. And I thank you for your attention. And I'm happy to answer for some questions.
Thank you very much. So it's time to open the discussion. Now, all the members of the jury. So starting with the review of us. And the first review of us, I propose the podcast to start the discussion. You didn't share your discussion.
Microphone is not open. Proceed.
Jocelyn, votre micro, il est éteint.
Yes, I was sorry, but I was just telling that I could not hear Mireille if she said something. I could not hear it. So
I said that the presentation was very nice for dance and rich of information. That was the moment for open the discussion with the members of the jury. And the first maneuver, I suppose,
Maybe Mireille, you can go on stage because we can hardly hear you.
No, ça marche pas là-bas. C'est ici. Here. So yeah, the idea is what Mireille was saying is that we started the discussion and the first is to Jocelyn to ask some questions.
Okay,
Want.
so I start. Thank you very much. So first, I thank you for inviting me to be in your committee. It was really interesting to read your document and to discover plenty of work that you did that I didn't know, of course, and that's really impressive. Your document and your presentation shows a wide spectrum of skills, scientific skills that you have, and the wide spectrum of collaborations, so on the worldwide experience. And what I'm very sensitive to is the long-range collaboration that you have with the Centre Eugène Marquis, and with a very deep involvement in these topics and clinical studies and collaboration with the oncologist and radiophysicist and so on. And that gives to your work a dimension which is really interesting, because you had access to many data and to many patients, and you used them in a very clever way. And also on the general side, I think it's also very interesting because in your presentation, we see that we do not have to choose between big data or models. You can have both. And in many applications, it's better to have both than to have only a single side of the picture. So that's really interesting. Maybe I can put a few questions on the different topics. In fact, I had, maybe it's a stupid question regarding the first part, more about the study of recurrence and toxicity, based basically when you get some deformable registration of people, I cannot really understand how is it possible to deform those maps, since the dose models are very complex. The interaction of the radiations, the X-rays or the photons, or I don't know what, with the organs is a very complex things with some strange curves for the absorption and so on, and diffusion and so on. And so I cannot understand how is it possible, in fact, just to take a dose map from one patient and deform it to another one based on the anatomical deformation. Can you explain me a little bit more?
Yes, Jocelyn, thank you for your question. Yes, it's a question that's been asked many times in a different conference as well. Actually, it's a very interesting. We tried to answer to this question at the beginning with the Gael and Eugenia as well. Well, we are not in an intra-individual context. We are not into a accumulated dose distribution where the format dose can have a physical interpretations, because we are accumulating energy and we're accumulating energy per mass. But the idea here is not to deform the energy or the dose to have well, you are not deforming we are getting around this interaction between dose, energy, and body, because the idea of this registration was to obtain just anatomical correspondences. So we consider a portion of an organ being irradiated. When you deform the organ, this dose is just interpolated, just extrapolated, and the idea is to have just an anatomical reference in order to compare the dose in a portion of the organ. So it's the same idea of splitting the organs and then compare the organs, the dose across the population. So these methods, of course, you are aware of that. These methods are inspired from these neuroimaging methodologies of voxel-based analysis, VVM, or proposed by Asburner and Freestone at the end of the 90s. And the idea was to study Alzheimer's disease. I worked in these methods in Australia. And the idea is just to find, in neuroimaging, they compensate by the Jacobian of the deformation field, because we are registering probability maps. And by compensating by the Jacobian, we compensate by this deformation. But here we are not having this probability mass after segmentation. We are having just a 3D dose distribution. So with a hypothesis that we are putting in front of that is that the hypothesis is that by deforming, we are not deforming the energy. We are just finding anatomical correspondences. So we are using a dose mapping. It was a spatial normalization. That's it. So the hypothesis is just that we are just achieving anatomical correspondences across the population. So, but it's a very interesting question, and we don't have any closed answer, cut answer to say why not taking into account. Right? Yeah, for the moment, it's not considered.
Okay, thank you. I've cut my camera because I have a very bad connection. I had to use the Wi-Fi since we did not use H or I don't remember number,
yes.
VideoConf, and so my connection has not a very good quality, so I cut the camera, turned it off, so sorry. Maybe a general question regarding radiotherapy. And in fact, my impression is that, okay, the machines have evolved a lot. The technology has evolved a lot, but finally, the way people compute the dose with the DVH, with the CT is really conservative. And do you see any hope that it will change? Because I was working in radiotherapy long ago, but I have the impression that on those points, it does not change a lot. Is it due to the company that keep their software very similar, or they don't want to take risk, or I don't know? And how would you see the future of that for future radiophysicists?
Well, it's a question for a medical physicist and radiation oncologist probably, but by being working with that, I think, yes, I think the DVH is becoming standardized because of the prediction that has been, well, the models, initial models were proposed on that. And also it's in the machines as well. The recommendations are the ICRU recommendation. All the recommendations are based on DVH. So the conservative way is to use the recommendation which comes from studies. The recommendations are not from the mind of a mother radiation oncologist. No, it's coming from several studies with different cohorts showing that V70 or every being in the history, the DVH is important, and the recommendation come from there. But it's not also the only constraint that you see in the machines today. You see the mean dose, or you optimize for some kind of constraint that appears in the machine. I think that the concept that we propose is a breakthrough. It's not the same thing as we are doing DVH like that. No, we are proposing methods that trying to first use a part of the organs as a predictors. So it's not the whole organ. The machines today, the constraint that you have in the machines, but probably I'm wrong in my answer. A medical physicist will say, what are you saying? But the idea is in the concept of splitting the subregions and use the dose to these subregions in order to define new constraints, probably based on small deviations as well, but because this concept of deviations is the cornerstone of the planning in radiotherapy, and probably the concept that probably in the future developments, in the future plugins. My idea, or what I have in mind, is you have the console, and then you import the subregions. You import for a particular treatment. You have a particular map of subregions, particular map of risk, and you try to be conservative, but not organ-wise, but subregion-wise, and then try to figure out whether you can pair subregions, trying to develop tools in order to optimize. So it's a multifunction optimization. So we can think on Pareto optimization. We have multiple different objective functions in order to see whether there's a compromise between different objectives, and each subregions can be a different objective, and so try to optimize in that way the radiotherapy. Well, to move forward or to go further from this DVH organ-wise concept. So is that a proposal? That's a view, and the European project goes in that direction. Is in terms of once you have a validated subregion or scheme of subregions, trying to see whether for a particular patient we can import that in the TPS and trying to figure out ways of optimizing the treatment based on constraints per subregion. But it's an engineering point of view. It's not a practical point of view of radiation oncologists and medical physicists who are in the bed with the patient, with the machines, and know everything about what is the historical aspect of this. So it's my humble opinion.
Okay. I have another question related to the methods which are used for segmentation and in your document, but in many papers in the same way. Okay, when you evaluate some segmentation method, you use Dice, Osdorf, and all this kind of stuff, and you increase from 0.82 to 0.84 and so on. And do you think that it's really a suitable way of evaluating the method in terms of the clinical impact that makes the difference when you skip from 0.8 to 0.9? Is it a difference for the clinical point of view? Do you see any way of having a more clever analysis on the consequences, in fact, of the segmentation onto the results after, due to planning and things like that, related to planning and things like that?
Probably you are referring to the document exactly. I described a method for segmenting the prostate from using statistical shape models. Probably is that yes, we propose a method, atlas-based methods and statistical shape models regularization in order to segment the prostate that I didn't present here I didn't show in the presentation. Are you referring to those methods? Or
not to this one in particular? But it's an example, yes. You segment prostate with neural net or any other method, and at the end, the only way to evaluate our comparison with the others on Dice and Osdorf.
Yeah,
and that's the point that I wanted to raise.
No, yes, well, the choice of Dice or Harsdorff distance is because of historically are the scores that are being used by the medical imaging community. And actually, it's the only way to understand the difference with the literature or with the previously proposed methods. So it's because historically, from the medical imaging methodologist, is the way to compare. Of course, the Dice score is not optimal because the Dice score is dependent on the volumes. The higher the volume, the less reliable is the Dice score because the sensitivity of the Dice score with respect to the volume is very high. So for smaller volumes, the Dice can be very low for small variations. Whereas in large volumes like the bladders, of course, the Dice score doesn't move a lot with the missegmentations. The hazard might be more sensitive to this missegmentation, but indeed, from my point of view, those methods are not reliable at all. It's from a medical physics perspective or from a clinical applicability, those methods, well, having a Dice between 0.9 and 0.92 doesn't affect a lot. It's well, in terms of registration either, it's the same thing, which is not the proper way to evaluate segmentation methods. Well, I think the trend today is going towards the open science. Open science means open data. So I think there is a reluctance in the medical imaging or in the clinical community mainly because gathering data is complicated. Gathering data is you have to go through different studies, costly studies. So release data, open data is a way to do it. Create databases where people can use it, can learn from those data, and then try to integrate communities to compare with not only these scores that are pretty oldish, and to try to figure out other methods for comparison. Well, it's, not in terms of segmentation results, but also in terms of impact on planning those distributions.
I can't hear you. I don't know if you stopped speaking.
No, I don't know. I'm still speaking. Can you listen to me? Hello?
Sorry, I have lost. Now, yes, I have lost the communication for a few seconds.
Okay, so. Okay. So yeah, I was saying that well, the Dice and the
yeah.
Go ahead.
No, I just wanted to say that since you have a very fine analysis in subregions and where there is strong consequences on the toxicity or things like that or recurrence, would be really interesting also when you have some method for segmentation to see which region are well segmented, and because if it is a region where there is no issue about toxicity and things like that, maybe you can accept lower accuracy in segmentation. Whilst if it's a region which is highly sensitive, you need to be very accurate. So maybe it could be really interesting to mix your kind of sub-region approach of analysis of toxicity to your evaluation of more basic image processing stuff. Don't you think so?
Yes, yes. But basically, you are right. And I forgot to tell you we try to do that with Eugenia's work in the segmentation of the urethra, where we came up with some original methodologies to evaluate segmentation of the urethra. You see, the urethra is a very tiny tube within the prostate, and about 5 millimeters diameter, 5 millimeters. And the idea is how well, we showed that the urethra is very important in prediction of some symptoms in urinary toxicity, and what is the impact of the missegmentation in the urethra. So yeah, we try to do that well, it's a paper in the Green Journal, where we try to figure out new ways of evaluating the segmentation of the urethra based on distances, on center lines, on portion of the urethra segmented around a center line is or considered a tube-like structure, and the potential impact that it has in terms of estimation of dose distribution as well.
Okay. Maybe a last question regarding the generation of CT using GANs from MRI. Why finally don't you directly try to generate those maps from 3D MRI and the CT of patients that you would have in your database? Because I know that there are some groups where people take some images and dose plan, and afterwards using, for instance, a Monte Carlo simulation for having a very nice dose map. And afterwards, they only take new images of new patients and they directly generate the dose maps using some CNN. And wouldn't it be possible to do that from MRI to skip, in fact, the generation of pseudo CT?
Technically, it's feasible. Technically, a CT image from an MRI is an image. It's a synthetic. It's a transfer of information from one modality to the other. The dose distribution is an image as well. So it's an image of energy. So technically, it would be possible to generate the distribution from the MRI directly. After I think the idea to go in 2 steps is trying to control between the MR and the dose distribution, to control with the CT the errors due to the image conversion, only to the image, and then the impact of the image on the final dose. But indeed, there is an impact between the original MR and the dose distribution. And if technically it's feasible, why not going from the first to the second? Well, I guess there are important considerations in terms of planned dose distribution, because you start including constraints, of course, in the machines based on density of the tissues. The computation of the dose distribution is based on the density of the tissues, which is provided by this synthetic CT image. So that's why we start in the CT, and then from the CT to the dose again.
And maybe it was, sorry.
Yeah, and then, well,
please go ahead.
Yeah, and then from the MRI to the 3D dose, yes, but then how to obtain the parameters of the accelerators, the beams, the way in which the accelerators delivering the dose. I think there is some other issues there. But why not? I guess it's feasible.
Yeah. In fact, the example that I mentioned, it was in brachytherapy. So I think that's very different, because the parameters is a positive distribution of the seeds, and therefore it's maybe more straightforward that in your case. Maybe just a last question before I leave you to the others. Regarding MRI to histology registration and use of the results, how do you compare your development to what was done in particular by Rouvière in Lyon? They have been working for MRI relation to pathology.
Yeah.
Is there some specifically different in your case as compared to their?
So the idea of registering the MRI with histology is actually to validate the observation from the MRI with the stains in the histology. With the works of Rouvière is going in that direction of validating with histology. The originality of this work, actually, I didn't mention here, is the immunochemistry stains as well. We included the C31, Ki67 with two markers of proliferation and the endothelial cells as well, which were very incipient at that time in 2014 when we studied this study, and where there were others. Anand Madabuchi in the US in Cave Western University as well. He was doing exactly the same thing. And other studies in San Francisco doing registration, and particularly the segment registration approach was original because we performed actually from the NDPI images, which are huge in histopathology. We started in a single level. We took the images from there. We segmented the urethra, the peripheral zone, and the central zone. We did it in both images, MRI and histology, and we registered using non-registration methods, using basically the same approach as we did in rectum. We performed the registration towards the MR. And then you have both in the MR. Then the idea was to make regressors between the stains, for example, the Ki67, the regressors between the features obtained in the MR and the histology output. And it was with a reduced number of patients. So that was the main limitation of that.
Okay, thanks. Maybe I will finish there to leave a question to the others. Again, congratulations for this work, and I'm sure that your European project will be very fruitful and very interesting. And also congratulations for all your workflows that makes the explanation of the method very clear, and how the things relate to each other very clear. So thank you, Oscar.
Thank you, Jocelyn. Thank you for your questions.
Thank you.
Yeah. Okay. Okay. Constantinos Marias is the next turn for questions. Your turn for questions.
Okay. So, congratulations for the presentation. I think that I agree with Jocelyn that I learned a lot from this kind of very coherent presentation, because I have to admit that usually when we talk about the work we have done the last 5 years, there is a lot of heterogeneity. But this, I was very impressed how everything was like part of a common plan. So there were a lot of things that we could discuss. I will prioritize my questions that were generated during the presentation, and I think that one of the most important is the possible synergy between histopathology and imaging. And my question is, if you think, as an expert in the field, that this is something that can be further exploited. I know some things about the clinical workflow of prostate, but I think that you have, there is an ultrasound-guided biopsy that potentially that you could register to MRI. And this is during diagnosis, and also in cases you have prostatectomy, you also have histopathology specimens. So I wonder if the synergy between radiomics and pathomics, which is more, let's say, emerging, could facilitate some of the goals that you have presented.
Yes, it's something that I didn't mention, but actually it was we're working here in strong collaboration with the histopathology department in the hospital. Actually, there is some open roads for that. The first one is to improve prediction of recurrence and prediction of response to the therapy with a follow-up. Because you are right, we have been thinking in proteomics, genomics, radiomics, and then what is the room for histopathology? So we are thinking in how to improve prediction by including this histopathology data in the prediction. That's the first thing, is how to combine in our workflows using artificial intelligence, using machine learning methods in our workflow to find whether by using pathology with nice stains. I was thinking about Ki67 or markers of cell proliferation, or markers of endothelial cells with a indirect marker of hypoxia or marker of oxygen distribution. We might find some predictors of response to the therapy, complementary to the radiomics approaches. And these actually is far more useful, not only in the prediction, because it's an explanation, physiopathological explanation for what you are observing is something explainable. So that's the first window, the first thing that we are foreseeing. The second aspect is how to use histopathology as an observation mean in order to feed into the models, into the patient-specific models. So the idea is we have in the models, patient-specific models, this big problem of observability. We have a complex model of response to the radiotherapy that is, is giving a nice performance, but we still have a lot of parameters that are not observable. So the histopathology is, we are increasing, and we need more observables. We need more means of observability. To build a reliable model, we need more observable. The histopathology, I think, is giving us, or is giving the opportunity to increase the number of observable things in the single patient. So, for example, we can have access to cell proliferation, hypoxia markers, and to learn from how the response and how they are modified from a single patient, not only from prostatectomy specimens, but also from small samples. And then feed that into the models and build a more personalized model. That is the second aspect. And then run in silico simulations, but with more accurate estimation of patient parameters. That's for a moment the two things I see that can be useful in terms of joining digital pathology with MRI for the moment. Yes, that will be my answer for now.
Okay, yeah, I mean, some of the things I would have the same feeling. So, my next question, and I think the final one, because I mean, I can imagine that we have time limitations and for everybody. So, my next question in terms of prioritization is that I read, and today I think you also discussed that currently you coordinate a program that targets more, let's say, the clinical translation of these technologies. So and I think that many people in our field, especially, I mean, I know more about medical image analysis, but of course this is a wider one. We have been criticized that there is very little clinical translation. So my question would be, I mean, you obviously have a number of possibilities. Which one you think that has the more chances to really become part of clinical routine, and how you plan to achieve that?
Yeah, thank you for this question. It's a huge question in every conference, methodology conference. Is the question from the bench, from the bench to the bed, how to translate all these methodologies into the clinics. That's it's a big issue. We are in all the conference papers, the question of producing nice methods in order to improve the segmentation from 1995 to 96, and it's significant. Well, exactly. It's methods, and what do you do with the methods? The big challenge is how to translate that in clinical practice. What is the benefits for the patient? What is the benefit in terms of workflows? What is how it makes it easier the life for the clinicians in order to automatically segment structures? How to improve the quality of the provided service to the patient? And not in artificial intelligence, not only in the workflows for the patient, but also in terms of organization, etc. I think that for the works that we have been doing in the last years, and from the feedback that we have from industrial partners that we are collaborating with, or we are planning to collaborate with, I think, well, although the introduction of MRI in the clinics is important, it's a breakthrough. Here in REN, we will have the MRI LINAC in fully operational in 2023. Although the LINAC will occupy a very important place in the radiotherapy in the future. MRI is the future in radiotherapy, and there are methods for improving that. There are a lot of people occupying the field. While pseudo CT generation, well, there are a lot of people working on that. Even the industrial are doing that. The companies are already generating pseudo CTs, are planning based on MRI. The segmentation methods using deep learning are already there. Well, it's almost, the problem is how to markers. Well, I miss the word of the how to validate the use in the clinics. But I think the breakthrough in all these works for me will be the use of more accurate segmentation of subregions. The first thing is the certification of the patient combining those with biological data. That is original, and we are doing that in a maturation project, combining some markers from blood tests with those symmetric tests in order to certify the patient. That's very important thing, very easy to implement, and to convince that the radiological test with those is important. But I think from a planning point of view, that this idea of subregions in order to better plan, to better tailor the treatment will be a very nice contribution for the patient, because what we are proposing is we have a patient, we tailor the perfect treatment for you with a reduced toxicity. So instead of having 20% of risk of rectal urinary toxicity, we are decreasing your risk of toxicity by 10% or by 20%. So, well, I sign to go through this treatment, where I have a blood test, I stratify my risk, and then I propose a tailored planning based on this concept of subregions on the MRI, combining of the MRI. So I use the MRI within the workflow, and then to that. And the European project goes into that direction. Which constraints should I apply to this patient or to these subregions in order to decrease the toxicity? And the tools are there. So we have, at some point we are very close to bring that to the industry or to the clinical, in terms of clinical benefits, to show the improvements in terms of clinical benefits. The next time is to run trials and randomized trials, and then go again to the label European labels and well, all this workflow for the products health, in health. Yeah.
Okay, thank you. I mean, these were my main questions. So I think I'm covered. So thank you very much.
Thank you, Costas, for your question. Thank you for your participation. Thank you very much. So David, now is your turn according to the president of the jury.
Yes, thank you very much, Oscar, and well done for the presentation. That was very dense, complete, and that give us a very nice overview of all the work that you did in the previous works. I think it was very clear for us to have a good overview of the direction and the target that you want to achieve. It was interesting and very complex. I think the document that you sent us was very dense also. And if I may have a small criticism, I think it was not always easy to follow because there are too much things. So sometimes I think could be nice to have some short summary, maybe at the beginning or at the end of the, the chapter. But I really like in particular, the multidisciplinary aspect of the whole project. So I will right now. I don't know if we can say that in English, but I will kill the suspense. I think you show us everything required to get habilitation à diriger des recherches. You show us your nice scientific prediction. You have a lot of PhD students, more than 12, if I remember correctly. A lot of projects, international collaboration. So everything is marked on your document. So it's really, really nice. I want also to thank you and all your colleagues for the invitation. It's always a pleasure to follow the works of the group in Rennes. It's really interesting, and I'm always eager to see the next plan. So I have a few questions maybe for you to start this discussion. Maybe if we start by the prediction of the treatment response and the work that you present us on the predicting the response either from the DVH of the 3D Voxel-wise probability distribution. It's maybe a detailed question, but I've been a bit. I have some question about the dose normalization. I think you wrote in a chapter that you perform some dose normalization before gathering all the dose distribution for all the patients. Can you explain me a bit more what you mean by this normalization and why it is needed?
Normalization of the dose? No, I was probably speaking of spatial normalization, which is the technical term that I am using for instead of registration.
Okay. Okay, sorry, I misunderstood. So you don't normalize the dose; it's always the absolute value of the dose.
Yes, what we did, for example, in some patients that they were with boost, we summed up the two dose distributions and having a single the single planned dose distributions. So we don't have any normalization other than spatial normalization, which is registration. Yeah.
Okay, so maybe I misunderstood this part. Thank you. And there has been some discussion about the deformable image registration of the dose by Jocelyn, I think. You deform the deposited energy or the dose directly?
The dose directly. The grays are already deformed. Okay. So actually, they are not compensated in the transformed space. They are just interpolated.
Okay. And did you try to check if there is a difference if you first convert the dose into deposited energy and then interpolate the deposited energy, and then at the end, by using the mean mass of the common patient space in a way to get back the dose? Because my feeling is that you may have some uncertainty, especially in high gradient areas in the images. But it's a feeling, so I never checked that.
No, actually, yes. Historically, we tried at the beginning. There was a lot of questions about how to deform the dose. So as far as I remember, I think we with a master student, I think it was his name. I forget his name. Well, we tried to actually convert the CT into the density maps, and then try to come back to the energy of the dose. Instead of having the dose, having the energy.
Yeah.
But you need both. You need the CT because you have the density, and then you have the dose, you have the energy at the beginning. And as far as I remember, there was no any modification because it was linear relationship at the end when you have the deformed dose and the deformed. We were in the linear relationship between both. So what you obtain is a kind of proportional value of the dose. And in the dose Voxel-wise analysis, it didn't have any impact, but it was a long time ago. So probably now with the number of patients increased, probably it's worth having a look on that. Actually, that you mentioned the gradient, exactly. Indeed, that was one of the problems because, for example, the rectum is in a very high gradient area. The dose in the rectum is very, very high gradient, and that's why we had a lot of issues of interindividual variability at the beginning because the registration was not accurate enough in order to rely on them for the Voxel-wise analysis. So that's why we improved the registration, and we took care of this high gradient area. And actually, we proposed in Gael's paper, we proposed a new method to validate the registration, which it was not DICE, was not central line, was something that we called dose organ overlap. And the dose organ overlap is something that we worked here with Jean-Jacques Bélanger here in the lab in order to come up with this index. It was an index that combines at the same time anatomical misalignment with dose misalignment. And then the idea was to show that with this score, the misalignment in high gradient areas was important, and it was important to take them into account. So it's in this medical physics paper in 2016 that by Gael Rehan, we have this explanation of how to use the dose organ overlap. Actually, that was used afterwards by Laura Cella. As far as I remember, in some of her papers, she worked on those organ overlap as well, and she showed some as a score for validating registration. But you're right; it's worth having a look now on the energy part in order to see whether we confirm these results as well.
Okay, thank you, Oscar. Maybe a few words about more conceptual question. Still, when you try to predict treatment response or toxicity or whatever, it's a way from the Voxel-wise dose distribution. In a way, you try to find a space of the data where you can find a separation, easy separation of the patient with or without toxicity, for example. And that's the common way to go. But if I'm not mistaken, it means that the number of dimensions of the input space is quite large because it can be images or dose distribution, while the number of dimensions of the output is low because it's only a toxicity or not toxicity. So it means that there is a large chance, a large probability that you may have bias or fortuitous correlation. You know what I mean? If you have a large input space and a small output space, you may have a correlation. You may find thousands of correlation, but you don't know which one is correct. How do you try to deal with that?
Are you speaking about the multiple comparison problem? For example, where you have thousands of voxels and you are doing statistical tests and you are doing, for example, t-tests or non-parametric tests, and you are ending up having a p-value, and this p-value means that you have a p005, but still you have thousands of voxels, and then you have the multiple comparison problem.
It could be for that, or it could be also for machine learning or whatever.
Yeah.
Methods.
Yeah, you're right. It's a very, very important problem when you are doing voxel-wise analysis. And actually, what we've done, the first works were using only simple comparison, non-parametric tests. But actually, in Eugenia Milona worked on different methods to cope with that problem. She implemented the permutation test, that allows you to have p-values a bit more robust in order to well, we start with Bonferroni correction, dividing by the number of voxels, FDR. But actually, the papers that she published in the Red Journal were the annexes were with solving this multiple comparison problem with a permutation test, which the results suggest that by doing so, well, you have more reliable subregions, and there are actually the original simple tests are very large, gives very large subregions. Whereas when you are doing, are tackling the problem of multiple comparison, the subregions are a bit more eroded. Actually, it kills a lot of false positive rate. The false positive rate actually issues. And in concerning the machine learning, actually, well, if you are doing data processing, well, there are a lot of problems, the imbalance problems, then the overfittings, well, all these problems. For the moment, what we have done is when we are in presence of highly imbalanced data, to test synthetic oversampling techniques like SMOT in order to create artificial individuals from in the space of the features. By using SMOT, for example, you enrich your unbalanced class and try to then train models by using synthetic data. And this is in the training part. Then to test the model, of course, we are doing some kind of scaffold validation, but without having into account the problem of machine learning in depth. We haven't, well, we had for the moment enough data. We have enough data in some of the problems to tackle this problem in a more proper way. What we were exploring as well was to use ensemble learning and stacking as well in order to cope with the problem of dependency of the models, not only random forest or PLS or PCA or SVM, but trying to come up with this trend in machine learning, which is ensemble learning, which you learn from different models and to try to get the best from these different models in order to predict. So yeah, for the moment, I will answer that way, very conservative answer.
Okay, thank you, Oscar. We can move to the mechanistic modeling. Maybe this is an aspect I don't know a lot about that, but I was very impressed by the results that you show in the documents. And if I understand correctly, it's a collaboration and multidisciplinary works with another team in the lab. And what was exactly your part of the work in this task? Because there are some very biological aspects. I'm not knowledgeable of those biological aspects. I don't know if you are, or if you maybe work more on the how can I say it, algorithms aspect of the model. What was your task, your part of the task in this work?
Very good question. I was wondering today what was our part. Actually, it was a very initially the motivation for this work was to try to model just the response of the tumor to the radiotherapy. So we went with the biologists. It was originally a regional project, Canceropol Grand Ouest, with people from Nantes, François Paris, who are an expert in radiobiology. So they have all this analysis. They know very well the radiobiology of tumors and the response to the radiotherapy. So the idea at the beginning was to study the response to the tumor and to figure out whether we were able to reproduce tumor growth and response to radiotherapy with a simple linear quadratic model, which is the simplest way to model those tumor interaction. And then, well, we obtained the first results with a net logo, very simple model. Net logo to reproduce the linear quadratic equation. This is a simple. The first mechanism that we evaluated was oxygenation, which is just a diffusion equation. It's a partial differential equation that we have, and by integrating this partial differential equation, we have the response of the oxygenation. And then to model the tumor growth is just duplicating the cells, and then linear quadratic equation is just having a probability of response to radiotherapy. That was the first model in the logo, implemented very easily, very simple way. And then, well, Carlos arrived, and then, well, he brought a lot of expertise in mathematics, a lot of expertise in computer methods, and we enjoy of the huge expertise of the Sepia team, who've been developing these models for years. It's the beginning of the 2000s, where they are developing these models in different contexts, in cardiac models, and they are very far into the conception and the ideas of modeling, of the methodologies based on modeling. So, well, this library was developed in the early 2000s, well, to answer to this problem of different formalisms, different scales, different time scales, space scales, and different ways to integrate data in order to answer to this question, to predict things from a physiologically perspective, and having explanation as well. And the models, they give them ideas, ideas in order to probably to test different hypotheses, and then to try to with different devices, implantable devices. So this project in this team is very well exploited, very far, because the models gives you a lot of ideas, new ideas to test hypotheses, to test things that probably unthinkable from another point of view. So seeing that we have cross-fertilization, cross-collaboration within the teams, we thought that we could apply the same ideas, the same reflection to the tumor growth and to the problematic of radiotherapy. So Carlos arrived, and he developed in collaboration with this team. He took the library. He adapted the idea to this problem of cells, tumor growth and cells. So instead of having cardiac cells, we have a tissue, a synthetic tissue. And working with Nantes, we try to integrate all this knowledge in radiobiology. And our work was on testing, well, bringing together, it's a leverage of multidisciplinary things. We have clinics from the one side, radiobiology from the other side, computer science from the other side, the methodologies in modeling, and to put together this specific problem. But it's been a long road in order to come up with this model. Actually, the paper that was published this year is an IEEE transaction in biomedical engineering. It's a long story. It's been like 2 years of work, of waiting, writing, rejecting, submitting again. And well, once the paper was published, we see the road has been we've been through, and it's a long journey actually for this paper. And I think our contribution was to put everything together, to integrate a model applied to our problem of tumor growth and radiotherapy with multiple scales, enjoying the advantages of N2SL to integrate all these things. So it's basically our work was to integrate everything, to bring knowledge from different disciplines and to apply to the problem. It's a new problem to be tackled with these models.
Okay, thank you.
And actually, it was the implementation that we found for predicting the recurrence was afterwards. We didn't intend to do that at the beginning. We just tried to understand to implement the model. Now the application is straightforward. We can apply that for predicting, but we can apply that also to planning. So we are now foreseeing the huge potential of this idea of digital patient and planning based on models.
Yeah, I agree with that. And the answer was a long journey also. Still, sorry. Still, maybe one small question about the MRI part. I will skip maybe the part on the planification because we already discussed a lot on that. There are a lot of work on that. Maybe I just want to come back on one of your slides. I noticed it was the 115. You show us that there is some. You wrote that there are some differences in tumor contour size between the MRI and the CT, and I'm very interested in that types of observation. Can you comment a bit on why do you think there are some systematic differences in? You don't write systematic, but I understand that.
And yeah, that has been reported. Actually, it's been reported that you see different things in both modalities, and probably it leads to a overestimation when you segment the prostate in the CT and in the MR. There are huge difference. As far as I remember, I don't know if the number is high, but it's up to 20% of the volume is the prostate is higher. The volume is bigger in the CT than in the MR. So you can see the huge thing. We are doing IMRT CyberKnife based on CT, and you are targeting, and your target is bigger than it is supposed to be. So it's 20% bigger in CT than in MR. So by doing the planning in the MR, of course, you are delineating the thing that you need to treat, not overestimating that. And there's reference. I don't have the reference here, but there are a lot of reference that talking about this huge difference in observed difference in the same patients in prostate delineation.
Yeah, but all the experiences in radiation therapy, so the way the radiation physicist put the margins, the PTV to GTV to CTV margin are based on CT and some like histology study. So it means, maybe if I understand correctly, that we will have to redo in a way those study with MRI, because maybe you can see a smaller GTV in a way on MRI, but you don't know exactly how to adapt the margin for those patients, right?
Probably, yes. That has to be redo it. Yeah.
And do you have some plan with histology? I really like the work you did on the histology comparison between MRI, and it was some years ago. And you mentioned that it was unpublished, but I think there is a publication, but maybe I'm wrong.
No, you are right. It was a very nice work performed by Frederic Commander, who did a great job on that. But actually, it was with Roman Matthew at that period, and Nathalie Leclerc and the histopathology department. Of course, there is a huge potential on that. It was performed it was thanks to an ARC project, and we studied performed the study on 9 patients only. So we have data for 9 patients. We have registration for 9 patients, and actually the material is there. It's really unpublished. The registration part was published in an SB paper, but the results are not published. I consider, coming back to the histology, yes, of course, there is a huge potential in terms of comparison between the MRI and the histology to redo the studies in terms of margins and of course.
Yeah, but I understand it's a huge work. Maybe just a last comment before. You mentioned a lot of software that you developed, or you participate to the development of a lot of software for Voxel-wise dose prediction, a lot of registration software, etc. What is the policy in the group regarding the software? Do you want to patent the software? Do you want to open it? what is the policy for you and your future students?
Yes, yeah, it's a good question, David, because it's a question that methodologists are facing up every day. Whether we go to open source methodology, open source philosophy, or we keep the software for us and we only use it for two or three PhD students are going to use the software and that's it. Or we open it and we decide that everyone can use it and we can compare and we can do open size. Of course, I am more in favor of that option. Here what happened is what we register a software. We have the LASAT, this is the West Valorization, is the who is mandated, who has the opportunity of the grant is granted by has been granted by the university in order to protect, to do all these IP policies, IP protection. So what we did is to have software developed. For example, the software that we developed in the context of the LabX, InterLabX, RedTox, in order to import the regions to the TPS. That is a registered software, is being tested, and we are going to integrate that into the European project. So all the partners within the European project are able to use that software. It's very simple software. It's use a web interface in order to import, to generate DICOM-RT structures, and these DICOM-RT structures can be imported afterwards in the TPS. That is very simple as that. But well, it's registered, and we put it in the consortium agreement to be used by all the partners in the European project, for example. But if something happens, well, it's within the consortium, and if something happens, if there is an industrial interested in that, we'll have to pay for the license for the software. So for the moment, it's registered with here in protected by the LASAT. Yeah, so we have this policy of licensing, of registering software for that. The other software, well, they are some of them are very old. So the technology, we can do better things today with deep learning and other things, but the software is already registered. Probably is going to be used never. But from there, we developed further things afterwards. And in terms of tools, we have, well, since when I arrived to the lab, it's something that I worked before in Australia, is having like a common SVN tool that everyone is using, and we are bringing together new tools. So when a PhD student arrives, he doesn't have to reintroduce the wheel, reinvent the wheel, and reinvent the warm water. So they take the whole knowledge of everything, of all the students called LTC tools, and well, thanks to Jeremy Beaumont, who has been contributing and Pierre Fontaine in last years to structure that tools on that. So every PhD student arrives, and then it uses very easily. So it's in command line, it's in Linux, and you have to use well, it's platform that you use very easily. You can do batch in Python on Bash, or write your own algorithms, and then you do data processing on that. So that's the way in which we keep growing these suites of tools.
Okay, thank you very much, and again, congratulations and good luck. I'm eager to see the results of the European project. Thank you, Oscar.
Thank you, David. Thank you, David, for your questions. Laura Cella? Yeah, okay. Laura.
Hi.
Laura. Hi, Laura.
Hi, Oscar. Congratulations for your work and for your presentation and for all the pipeline, because everything is connected, and so this is great for, I think, for your career. And I have only one question, and my question is, for all the Voxel-wise analysis is based on planning those maps. And what is your position about organ movement and the possibility of using accumulated doses instead of planning doses? This is my question.
Very, very good question, Laura. Thank you very much, because it's a cornerstone question. It's the question. Actually, the doses that we are using to compare across the population are planning those distributions. And well, actually, the dose distributions it's been shown that the planning dose distribution is not the same as the delivered dose. So where we are achieving the same results or having the same results with the planning dose distribution, or instead of using the real delivered dose, is an open question. And of course, it has to be done. The delivered dose, considering all these problems that we have been talking about before, in terms of overestimation of organs, of course, what we have to take into account is the real delivered dose and fractionated accumulated after the treatments. Definitely, this has to be done. There's a nice paper that Leila Shelley in UK, who did this nice analysis using mechanistic models, trying to compare the delivered dose distributions with the planning dose distribution. And she didn't find any huge difference in the rectum. There is nice works from Claudio Fiorino showing that the difference between planning distributions in bladder and showing that in some parts of the bladder there is no big difference as well. There is here in the lab there is some works already done in terms of comparing the planning dose distribution with the cumulative dose distribution in rectum and in the bladder as well, and studying the impact on the using cumulative versus planning dose distribution using different strategies, registration and use centered on prostate, etc. And actually, it has to be done in terms of toxicity, whether we find the same regions or not, whether the regions are validated or not. I expect that, well, in some organs not. Urethra it will be okay. Probably in the rectum, in the high-range dose, will be some impact, as well as in the disease of the bladder, probably not, but probably in the trigon as well. I don't know. Well, it's a good question. It has to be answered, of course.
Perhaps a good approach could be to, especially if we would transfer all this knowledge in the clinical setting, is to imagine a safety margin on the, like the PTV, CTV, also for the substructures. So say we have a substructure plus minus a margin due to the organ movements. Because if you think to use this substructure for planning, we have to be sure that there is the right place we are considering.
Yes.
Something like this, I think.
Yes, it's a very good, of course, it's an unavoidable question.
Okay, I'm done.
Thank you, Lara. Thank you, Lara.
Thank you very much.
Caroline Petitjean.
Okay, thank you. Okay, so Oscar, thank you for the presentation and congratulations for all the work that you have accomplished and all the contribution to the community, the methods, the software, the publication. It's all very impressive. And also congratulations to the nice recent achievement, the ISB co-chairing, the large-scale European project that you are leading. All this show that you are recognized and well-established researcher in image analysis to improve the radiotherapy planning. So I have to say I remember your work early in multi-atlas segmentation in the beginning of the year 2010 that I was very interested in, and it was really a good contribution to me. And but it was just the tip of the iceberg, apparently, because I was happy to discover the rest of the iceberg in your manuscript that I didn't know, and I learned a lot of things like my colleague, and so it's very nice that we have an opportunity to discuss today. And so I would like to also say that I enjoy reading the manuscript and especially the workflow that you draw. I think it was said by by several colleagues earlier that you really know how to synthesize complex workflows and concepts. So it's a good point for that. And at last, I would like to thank you for inviting me to your HDR jury, because I'm very honored. It's my first time in a jury of HDR. So thank you. Maybe my field of interest is more the methodological aspect of image analysis. So my first question will be on deep learning, how it has impacted your research, because I've seen that you've been using it for a specific part of your research, especially for the generation of synthetic CT images. So how did it impact your research, and is it for you a revolution, or is it just another tool? Is it interchangeable with the previous techniques? And okay, so this will be the first part of my question.
Wow, this is a very deep question.
Okay.
because actually, yes, you are impacted. All the community is impacted by deep learning.
Yes. Mm-hmm.
Because well, is neural networks, and the concept is like artificial intelligence. These concepts are date from the 60s, 70s, and neural networks as well. So because we have today the computational capacities, abilities to implement these nice ideas, well, we have seen the potential, and actually, well, but it reflects also the way in which algorithms can work in other ways. I mean, it's taking diffuse data and by doing aggregation of knowledge, or partial knowledge, coming up with decisions. And segmentation is that you are doing convolutional networks and then trying to figure out whether we can infer some knowledge from this locally, but also contextual information to come up with a decision of whether this is a segmentation or not, or whether this a patient is being classified or not. I mean, the opportunities are huge in terms of segmentation, registration, predictions. And we show only one example with the GANs. With the is very nice implemented, because well, it was Axel's work in a few years ago, 3 years ago now. He implemented this GAN, and he showed very nicely how to transfer an MR and to convert an MR in a CT. And well, you just open, you see, we are seeing only the peak, the tip of the iceberg, because this is opening a lot of opportunities for us. Afterwards, it's a bit of hassle to have this deep learning everywhere. You see, well, you're more than me in the MICAI's conference, BSB, how deep learning has invaded everything. And now we are about what is the best loss function, what is the best architecture. And actually, for example, in the European project, we leave this part of segmentation using deep learning to the Spanish guys, because they have already developed nice algorithms for using deep learning in segmenting organs at risk, mainly head and neck. Now we are transferring that to we have nice results actually in the European project on using this data, using deep learning segmentation. We are segmenting the prostate, we are segmenting the urethra with deep learning already, the bladder and the rectum, and the results are coming up, are very nice. And well, we have to do width. We have to go through that. And actually, the sad part is that we have, with Frederic, we have been working in some nice statistical chain models for segmenting the prostate. And what to do with that? Those methods based on statistical chain models, on spherical harmonics regularization, nice mathematical methods, nice methodologies are useless now or not? What is the future of that? We had nice results on prostate segmentation. We have unpublished results on that. And those methods, unfortunately, they last forever. They last for 40 minutes, 50 minutes to segment one prostate. They are useless in clinics. Now, with deep learning, you have a result in a few seconds. So the problem is how to segment. Well, there are other issues in terms of optimization and validation in external cohorts. But actually, well, all these traditional methods are basically, well, are there, but we don't have to use them anymore. One day, when the computational capacities are increasing, probably we'll have these methods coming back. Yeah, will be my overview of deep learning. But it's nice. It's nice. I mean, I have to confess, it's very, very nice to use that now.
I agree with you. It's as you said, it has both side. The performance, the good performance side, and the hassle of having to do it, because otherwise you are under the radar on the methodological aspect of the analysis. So just my last question regarding deep learning, because you've worked on radiomics and on specific image feature extraction. So do you think, because with the CNN you can learn the representation, you can learn the feature and not decide a priori, you know, to learn to use heraldic features of this kind of thing. So I don't know if you are still working with the radiomic and image feature extraction, and if it's good for if it would be useful for prediction, for recurrence prediction, to use deep learning features. Or
actually, it's a very question, Caroline, because my heart is split in two. Because first thing, I think the deep learning has replaced everything, and although it has replaced everything, I don't see any future in radiomics by extracting handcraft features. The handcraft, you are like guessing with the hands. You are looking whether this feature is correlated with something that is, well, like a 5 years recurrence. And then, for example, you might find that some heraldic features are correlated also with, I don't know, for Gabor filters or something like that. Mm-hmm. And there is no relationship. You are like, it's handcraft feature. And then you predict, you find that something is predictable. But actually, this approach is you are in a blind world. You don't know what are you looking at, and you are extracting some features, and you find that by randomly, you are finding correlations. Okay. That's perfect. It's like the bioinformatics with the ADN or with the genetic code, but you are finding something that is not interpretable. And this is the big difficulty in radiomics. And for me, deep learning came around to replace a bit that concept of handcraft, because it's learning local correlations, contextual correlation, is multi-scale. And by being multi-scale is learning all the things to predict, and it is more elegant to do it that way. And the other question is about we are in the Pierre Fontaine's PhD work with Adrien de Poursange in Switzerland. Adrien de Poursange has been at the heart of very initiatives in radiomics. What he's been shown, Pierre Fontaine is not published work yet. Well, yes, he published a paper in Scientific Reports this year. It's about showing the heterogeneity of the delineations in terms of radiomics predictions. So by doing some methods, aggregation methods, or by looking at the heterogeneity of the tumors, actually what he's shown is that some radiomics feature might be not predictive or not, but it does depend on the feature. It depends on how do you perform the segmentation, or which areas are you performing the segmentation, or how you do. And I think there is still some room in order to improve radiomics, but studying at the basis. What are you extracting? Are you extracting the tumor activity, the heterogeneity, or how to aggregate these features in order to be more predictive? So yeah, we are still working on that, and thanks to Pierre Fontaine's work, and I live with Adrien de Poursange. I think I have both questions. I'm not closing everything. I'm not closing the doors of radiomics, neither the deep learning. I think we have to keep mind open.
Yeah, exactly. And you addressed the question of explainability, of interpretability of the models, and so yeah, it's a true question. And your mechanistic models that you showed in your presentation was a good example of a true interpretable models, contrary to deep learning models, and a lot remains to be done in this area for CNN. So. Okay, let's close the deep learning door, if I may say like this. Now I have a question about all the images that you are working with, and because you are working with the clinical department, and you have some Renaud de Crevoisier or some other medical expert with you. So how is it to work with the new regulation RGPD law? Is it still easy to work with the images coming from hospitals? And do you think that you can make some of them public some days to organize a challenge in your field?
Very, very good. Actually, these RGPD things are the first thing that when we are starting the European project is to fit with. I mean, in the data management project, we have to include all these RGPD rules, and of course, it's a big issue for us to work with that. I think we have to go through that. in the benefits of the patients, in the protection of the data, the protection of confidentiality, the protection of, well, you have to go through this regulation, otherwise it will be a mess in terms of data exploitation. I mean, you can take data from a study performed a few years ago, and then you don't care about what it was intended for. So you use it the first time for I don't know, something specific, and then you find the data, use it by another lab in another thing, and then you use it for another different lab in another thing. I think that must be the regulatory part must be respected. It can be a hassle to go through this RGPD thing, but I think it's necessary in order to keep put order in everything. I mean, stop with all this mess of changing data with the USB keys, and then let's go through something more protected in terms of confidentiality. And after that, I must say that I said before that I think the clinicians are a bit reluctant in order when it deals with open data aspects. Mm-hmm. And I think it's important to go through that. It's a kind of it's hard. It's hard to go through that. We can learn from the neuroimaging domain. In neuroimaging, today we speak about ADNI database, for example. It's huge amount of data. Mm. And at the beginning, the images for Alzheimer were very close. But in the early 2000, where we open everything, we're speaking about ADNI, thousands of patients, and thanks to this open data philosophy, new methods emerged, and we could finally test across the methods with the same data, and that is important. Have the same data and have people sharing the data in order to test different methodologies. Mm-hmm. And it's the challenge in MICAI, for example, by going in that direction. Adrian de Pour-Science is organizing this challenge in PET analysis as well. Well, those data are coming also from here. We have Joel Castelli working in this project. Open data in order to test different methods to predict survival and to predict segmentation as well. So this is a real question, and I will be willing to work in initiatives in order to release at least partially some data for the communities, for the medical imaging communities. Otherwise, our methods doesn't help a lot. Won't help a lot.
Okay, thank you. And regarding your European project, which is very impressive, and I think it has the potential to make a great impact. Ultimately, its goal, is it to make some recommendation? I mean, the outcome of this project, will it be to make some recommendation for treatment planning, or I mean, this is more a general question, how your findings can be translated into the clinical world for treatment planning?
Well, the philosophy of the ERA PERMED is personalized medicine. So the call was around personalized medicine. So the idea was to have already methodologies partially tested, partially validated. Mm-hmm. And the goal for the ERA PERMED was to bring up some methods that will be close to the clinics, close to be used to the clinics, or in phase of validations. So our idea in this project was to use those voxel-based models approach already to go to real patients. So the idea is to find ways, to find paths in order to bring that to the clinics through the implementation of two things. First, external validation, and second, to develop tools in order to transfer directly to the TPS, in order to plan directly with the subregions. Mm-hmm. And that's part of the Italian expertise, is to bring to develop methods in order to test different configuration, different optimization methods, in order to see whether by doing these constraints on the subregions we can achieve a better prediction, we can reduce the toxicity, and then do that by simulation, by simulating on different cohorts, testing, simulating, and optimizing the loop of the workflow for radiotherapy. That's the idea. So we have to go close to the clinics. We have to go close to the patients, and the European project is intended to do that, to bring tools in order to achieve a better planning.
Okay.
That's the idea. Better prediction and better planning.
Okay, and last question, just to learn one more thing with you. What is MRI LINAC? Because it's the first time that I hear about it. So.
Well, basically, it's bringing the benefits of both the MRI and the accelerator in the same setup. Usually, the accelerators were well, the evolution of the accelerators were based on CT. And for the first time, we're going to have an MR in the room, and that is able to at the same time have images and MR images and the accelerator compatible with the accelerator, linear accelerator on the same patient. So the patient will benefit. In the workflow, we can introduce the knowledge that we have on MR and the accelerator. So it's a treatment planning, it's a treatment system based with an MR inside.
Okay, so it's not widespread in France. We cannot find MRI LINAC in every city.
I don't know. I think there are Renault de Croatia is raising three fingers. So I think there are three accelerators currently in France, and in Rennes we will have one in 2023.
Okay.
So his cost is very high. We are talking about 10 million euros, the full facility. So because of the cost and so the bet is that for me the bet is, well, I don't like to be attached to a given technology because there are a lot of risks. But the bet is that if we have the accelerators, the MRI LINAC in 2023 fully operational, the opportunities in order to improve the field will be huge for us in terms of transfer of all this knowledge to the clinics.
Yes. Okay. Well, it will be very interesting to follow, and Rennes will be the place to be on this topic. So good for you, and congratulations again. And I will give the speech back to the president. I don't know if we say that in English, but
thank you, Caroline. Merci. Donc Renaud and Mireille are coming to the on the spot. Are going to be on the spot. I leave the room for them. Yeah, the micro is here.
Okay.
That's this one. So Oscar, today is a big event. Huh? It will be up to the, of course, it will be up to the examinators and to the members of the jury to decide. But you made really a huge work, which may represent something like 2 or 3 HDR. So congratulations for all this work. It's really a pleasure to work with you. I want to stress your investments, your enthusiasm. We spent, I don't know, 100 hours together working on the manuscript, trying to improve all our work. It's really a very nice adventure, which began actually something like 13 years ago with you, with Pascal Aigron, with Antoine Simon, and then with Jean-Claude Nunes. And a very nice adventure to work all together and develop translational research in radiation, as well with the physicist of the Centre Eugène Marquis. I'm thinking about Caroline Lafont, about Anaïs Barato, all very nice people. And I want also to thank Lotfi Senaji, who trusts also all the team and support the team to develop all this nice work. And I hope you will continue to work at, I'm sure that you will do, and through this nice project and other projects. And again, thank you very much for all your work, your very nice work, both in quality, both in quantity. I'm not sure that somebody can do more than that in this in all this duration.
Thank you very much. So First of all, I will have some words for you, Oscar, first of all, and after for the members of the jury. So I'm very happy to see you defending your health here today. Yes, of course, for you, for the team, in fact, and for the lab. I would like, in addition to what Renaud has already said, maybe I would like to recall a little history. And the path that brought you to this situation today, with the richness of your collaboration, local, patient, but also international level. And so, thanks to your dynamism, I think, scientific and human high qualities that you have. You arrived, yes, as you have said, in Brest several years ago, and after you have the opportunity to come in the lab, where we were very likely to welcome you after the PhD, and after doing 5 years, so you have passed a long time in Australia, and where you were enriching your activities and your collaborations, and you were able to build a network of rich collaboration, very solid collaborations that you have already today, and while always developing also collaboration with Colombia as your home country, and that you are extending with now other collaboration with Switzerland and so on, and so very rich collaboration. So not only have you been able to build up this collaboration for your work, but also to benefit the doctoral students, but also your colleagues in the lab, that you have been able to involve in this dynamic. So it's very nice, very important. You were and you are also one, I think, the pilot member of in the lab that made the activity of the radiotherapy grow up, and with now other colleagues. So it's nice. You succeeded in harmonizing, I think, a whole set of coherent activities in this field. In the center of multidisciplinary team, as it has been said, with Renaud and the team in the Angel Marquis Center. You also showed an ability to lead inter-activities, inter-project activities, with CPR, for example. So it's we see that you have the ability to energize and bring together. So it's very good. So many thanks to you for all that, and all that you have built and that you will continue to undertake in the lab for us. Now for the members of the jury, so I would like to warmly thank you, all the members of the jury, for their work, their participation today, and for all the remarks, very rich, for the work of Oscar, and for the work for the lab and the team for the future. As it was said also by whole Oscar, so you have showed largely your capacity to supervise young researchers, and also undergraduate and graduate students, and to lead to them on innovative topics, always with a clinical purpose, and to produce very nice results. So you have presented very good prospects, especially with the European project, and so for which you are responsible and the principal investigator. So it's very nice. So in conclusion, we can see from all this discussion the richness of your work and the extent of work you have undertaken all these years, and it's a very big work. So all my congratulations for your work. Thank you.
Thank you, Mary, for your kind words.
So now I think if there in the members of the jury, if you have another remark or question, is also possible. If you have, we can take the question. No. In the room, is there any question? No, no remark. So I think we will, if there is no other question or remark, we will finished discussion now, and we will be able to deliberate in this room. So
what I'm going to do is
thank you for Paul to be here, and we will continue here.
Okay.
Deliberation.
So I just got the micro for a while.
Just to say that to everyone in the jury, so we will you have to connect to the second link, Zoom, and to discuss together with my computer. And so you can disconnect here, you connect in the second link, and then so you can come back here in the first link.
Okay. Huh? So I just let me put that into I
Yeah, again.
Back. In which town, in which city do you are?
Napoli.
Napoli, okay. This house.
Yes. And you are?
In Lyon.
Yeah, okay.
We have a PhD student that came from Napoli, so.
Ah. From which department? University? No.
I don't know. Sorry.
But this is in which specialty?
Biomedical.
Engineering.
Là, vous êtes là. Miguel est en train de démontrer ça. Non. Là, c'est bon. C'est avec là, vous voyez pas. Là, vous voyez. Miguel, on voit. Ils vont vous voir. Là, on va voir. Attends, ils vont vous voir là-bas. Comment? Tchum. Et là? Ils sont là. Voilà. Voilà, ils sont là. Oui, regarde. Tu as assez d'accord. C'est là.
Oui. Tu vois, c'est pour qu'on voit mieux tout le monde. Attends. Voilà.
Ready, Oscar?
Oui.
Ok. Reste dans la caméra. Oscar?
Ah.
Je veux pas.
D'accord.
Non, mais là. Oscar, so the jury appreciated your presentation, the high quality of the scientific presentation, with a nice overview of the work that you have conducted all these 12 years, during all these 12 years. You have shown the multidisciplinarity of your activity that is rated from computer science, medical physics, and clinics also, real application in radiotherapy. You have shown also that you have a great rich network of international collaboration today, and the main accomplishment of the work is a high number of publications, a high number of projects, the ability to find money, and now the European project, for which you are the principal investigator. The jury is impressed. Is impressed by your long-term vision and the fact that you keep this vision along the years and along the work, and that is very remarkable. About the discussion, so you have answered to a large number of various questions from a high multidisciplinary jury, and the responses have shown also that you are an international expert of your domain, your field. You have also aspired a lot of thoughts in scientific topics. And so all the presentation and all the responses of the questions have highlighted the high quality of your work. So all these elements justify the ability of Oscar to direct research.
Thank you very much.
Congratulations.
Thank you very much. Well, thank you very much. Thank you first, Mirai, for this kind words, which is, I guess, represents the opinion of the jury. And I deeply appreciate this, the special care of each one of the work, because I think I understand that I infer that there are a lot of not only rationalized scientific aspect, but also emotional part. So I really appreciate this as a part of, well, I think it from my heart directly. And well, thank you, Renaud, as well, for being here for that. Well, I come back to you. First, also thank you to all the members of the jury, first of all, for being here, for your feedback, for your questions, for your kindness and amenability in the feedback that you gave me. I think that you gave me a lot of vision as well, because I take in all of your ideas in order to improve some of the things I wanted to develop as well. Thank you for that. Thank you for the time you spend, and not only today, because I know that it's huge time, taking all the whole afternoon and a Friday afternoon in July, in summer, some in Greece or in Italy or in Rouen or wherever you are, in Grenoble and Lyon. And I appreciate that you took the time to be with us here today. And thank you for the time that you spent doing the presentation, but also reading the document. I hope working with you in the future. I know that with some of you, we have things to do in the future. I know, well, Laura, I hope visiting you in Napoli and Costas in Creta as well in the future to do some, well, to expand this international collaboration, because it will be a real pleasure. And here in France, with David, Jocelyn and Caroline, merci beaucoup pour vos commentaires et vos remarques et qui sont très appréciables. Thank you, well, Mireille, for being the president of the jury. I appreciate your work, your introduction, very kind and very deeply moved for this work. Thank you, Renaud, because not only for being today, because of these 13 years of work. It's been a long journey, as I said before. I remember with Renaud, we met the first time in sharing a coffee, and he explained me in 5 minutes the radiotherapy, and I say, there is something to do with that. So indeed, there was something to do with that. I appreciated every moment, every deeply discussion with Renaud, not only the scientific part, but also as a person, as a human. I appreciate a lot. I remember we spent 2 weeks in Australia. It was a very nice trip to Australia. We visited 3 labs in 10 days. We visited Perth, Adelaide, and Brisbane in 10 days. And we made the tour of Australia, talking about collaboration, and we came back to France with a nice scholarship for Hilda and some grants, with a lot of ideas, exchanging with them. I spent a lot of time. It's Renaud who introduced me to the friends in Italy, Claudio Fiorino and Tiziana Rancati, who are the main partners in this project, the European project, that are today are not only collaborators but friends, because we have been spending time in the conference, in dining, in having nice conversation, but outside of the work as well, which is part of our work as a researchers, not only scientific part, but also the human part. And I am enjoying taking the opportunity to speak about the human part, because this work that I presented today is not my work. It's, as we discussed before, is the work of every PhD student, every master student has been collaborating, contributing on this work. Well, you will recognize yourselves when I am speaking. It's been the consequence of fruitful collaboration, and you know who I am speaking with. It's really nice being working with every single PhD student that's been co-supervised in this work. It's your work. It's not my work. It's your work. And because every development, every code, every line code is not mine. It's yours. And the only thing that I tried to do was to put people working together. That was my job in this. Thank you, for to all the colleagues in the lab. I speaking about the impact team, because first Antoine, Pascal Legrand, the first people that I met when I arrived to the lab, who introduced me to this world of radiotherapy, and I really want to thank them, because they opened the doors for being integrated in the team, and thank you very much for that. and I think there are things going on in this area, and thank you for this fruitful discussion as well, because mainly when Antoine, we've been exchanging a lot of ideas, things all the time, and the things that I am presenting here are also yours. So thank you for that. And now with Jean-Claude and all the people being integrated in the team, thank you very much, and all the members of the team, Impact team, well, like thinking of Jean-Louis with Miguel and all the people that are in the team we are working with today. And to all the other colleagues in the lab, well, of course, Lotti, because for your trust, your trust me since the beginning of this work, and he's all the time supporting us. And well, not only me, but all the masters, all the students from the PhD. Lotti has always the door open for everyone. Either you are a PhD student, a master student, or a researcher, or well, the door is always open, and it's very nice to have this kind of support of all these ideas for signing papers, for supporting things, for going further, for supporting us. En français aussi, pour nous supporter. So this is very nice, and all the colleagues in the lab, of course, all the teams. I can't name them, all of them, because they will recognize in my words. Well, of course, the ones that are here, thank you for being here, Virginie and Regine, of course, that are my friends as well. Thank you to all the medical physicists because this is something that is multidisciplinary, and is the work of well, without this multitask, multidisciplinary team, it's impossible to perform research as is shown here. It's definitely it has to involve engineers, radiation oncologists, clinicians, and medical physicists. Is synergy between different fields, and otherwise it's impossible to perform a good research, good publication without the impact that can have with the participation of different teams. And well, thank you to all the members, of course, of the lab. I think, you know, Fabrice and well, Miguel, thank you for your help, support today, because otherwise it would have been impossible to go through all this presentation. We've been through tests the whole week, and thank you, well, Miguel and Fabrice. Fabrice has been a friend for a few years now. He went to Colombia to my wedding a few years ago, and I think this is more stressful than a wedding. I've been and thank you for your support on everything, on these years of friendship. I'm thinking on all the friends in the lab that are not here, Alfredo and all the directors of the team. I'm thinking a special thought for Fabrice, who's taking over the lab in the next few years. I wish he could be here with me, because well, it's been since the beginning welcoming in the lab. And well, thinking back on the PhD student, I think now those are not PhD students. Those are my friends, my colleagues, and I think very often I am writing them back again to ask for some advice, because I think some of them are in very nice position, and they are more wiser than me. So I am writing them, calling them all the time. I have they became friends for me, and a very important part of my life. All these students that they went passing through the lab. Paul, je passe en français pour remercier tout le monde déjà. Donc, j'espère ne pas oublier personne, tous mes enfants qui sont là. Et ma famille, donc Alma, Emilio et Olivia, je suis très très content de les avoir ici. Je parle en français et je suis très très fier qu'ils soient avec moi. Et j'espère qu'ils ont bien un petit peu ce qu'ils faisaient papa. Et je pense aussi à tous les gens qui sont en train de suivre sur Internet, YouTube cette transmission. Ça nous ouvre la voie sur pas mal de choses. Et merci pour les messages que j'ai reçus très sympas sur YouTube. Ils sont vraiment, j'ai apprécié vos mots. Et tous les gens mes amis qui sont pas là, qui m'ont appuyé pendant toutes ces années, et ma famille qui sont un peu partout dans le monde, qui sont en train de suivre pareil. Et je pense à mes parents qui sont en Colombie en train de regarder, mes frères, mes sœurs, et ils sont un peu en train de regarder ça aussi. Quand mis papas, entonces un saludo muy especial. Es como les Tour de France à la fin, en fait. Merci a todos. Hola, gracias a todos. Entonces, gracias a todas las personas que me han ayudado en estos años. Gracias a todos mis amigos que están en Colombia siguiendo, a mi familia que está tal vez siguiéndome allá en Colombia. Les agradezco mucho su participación, su apoyo, su ayuda. Agradezco una palabra muy especial para Rocío, que me ha soportado ha tenido mucha paciencia conmigo estos últimos meses. Y también para todas las personas que están de alguna manera u otra vinculadas conmigo. So to close that, I think I'm a bit dry. So in order to continue discussing, I would like to invite you. Merci, Patricia, j'ai oublié de dire ceci, Patricia et Mireille qui ne sont pas là, qui sont toujours, et merci pour le support permanent sur tout, surtout, pas seulement dans les parties opérationnelles, mais aussi d'amitié, d'écoute, tout le temps les portes ouvertes. C'est quelque chose que j'apprécie au labo, c'est toujours cette chaleur qui existe au laboratoire. J'étais très content toutes ces années de pouvoir rentrer au labo. Il n'y a pas un jour où je sois malheureux de rentrer au labo et qui dit que je veux pas. Non, je suis trop content de venir au laboratoire travailler et je suis très content de continuer à travailler tout le temps, mais avec cette chaleur avec laquelle on est. Donc, pour continuer, to close that, I wish the jury could be here in order to have a drink, to share a drink, because I have some special food from Colombia. It's a shame that you're not here to share it with us. But actually, we are opening some bottles and we are going to have a very lively moment all together here. And donc je vous invite au club des professeurs. On va avoir quelques bouteilles, on va boire un petit peu, manger un petit peu et puis partager un moment plutôt décontracté maintenant parce que là, la pression est descendue un petit peu. Merci beaucoup à tous et à tout à l'heure.
Au revoir.
So I'm closing I don't know. Thank you very much. Thank you very much, Costas. Thank you very much again.
Bravo, Oscar.
Thank you, Oscar. Congratulations. Enjoy the evening.
Thank you.
Bye.
Bye, bye.
Bye. Congratulations.
Bye, Laura.
Bye.
Thank you very much.
And you have to send us the Colombian food.
You have to come. You have to meet together so you can be here to share this Colombian food.
We hope in the next future. Very close.
Yeah, bye, Laura. Thank you very much. See you soon. See you in Madrid. See you in Madrid for esto.
I don't know. I don't know if we are allowed.
Yeah, I already have the ticket, so you have to fly.
I have not taken the ticket yet.
Okay. I'm going to. Bye. Thank you, Costas.
Keep in touch.
Keep in touch, Laura. Thank you, Costas, for your comments as well. Gracias. Muchas gracias, Costas.
Chao.
Felicitaciones. Hasta pronto. Chao.
Gracias, Costas. Hasta luego. Adiós. Bueno, adiós, hasta pronto todos los que están conectados en línea. Adiós. Olivia, ¿quieres venir acá a decir buenos días? En YouTube. Vengan aquí en YouTube. Oui, vengan aquí en YouTube. Venez à YouTube. Vous faites coucou et on ferme. On va pas être en YouTube.
De toute façon, c'est que les gens.
Voilà. On va pas être là.
Mais c'est décalé.
Non, regarde, c'est un décalé, mais.
Ah oui, c'est décalé.
Voilà. Alma, Olivia, quítate la máscara.
Ah, ok.
Milo, quítate la máscara.
Ça va, coucou. Mais en fait, on voit pas. Attends, elle est où la caméra?
Mira, está, mira, digan allá. Ah, súper. Quítate la máscara, Milo, quítate la máscara.
Je l'ai pas. Regardez. Moi.
La cámara está allá, mira, digan allá. Alma, ven aquí, ven aquí.
Ahí là.
A ver, yo ahí. Listo. Bueno, ahora sí, adiós, adiós. Todos. Di adiós. Allez, on ferme. Arrête la diffusion. Dis-le, dis ciao.
Ciao. Hasta la vista.
Hasta la vista. Adiós. Vamos a tomar a comer unas empanadas y tomar unos traguitos. ¿Ok? Ya, vámonos. Chao. Adiós. Arrête.